I guess let me just position it.
There we go.
Okay. Are these videos coming out a little better now? I moved position a bit. Okay, good. Maybe you can even see a few things on the board. All right, what we got here? First of all, okay, first thing we want to look at is a gene called the tropomyosin gene. Now, what is tropomyosin? Tropomyosin is a protein that's mostly alpha helix, so it's a long extended molecule. And what it does is, if you remember a microfilament, it's like two beads that are forming a spiral coil, two strings of beads that are forming a spiral coil around each other. The tropomyosin fits in the groove between the spiral cords, between the two rows of beads. Now, what tropomyosin does is it stabilizes these actin microfilaments, keeps them from falling apart. In muscle, it also blocks the place where the motor protein myosin binds. It has to be pushed out of the way to allow muscle contraction to occur. And you get that if you take animal systems, phys, and stuff. But anyway, so tropomyosin, we have muscle versions of tropomyosin. They're found in things like skeletal muscle and something else. But all non-muscle cells and eukaryotes also have their own versions of tropomyosin. So there's all kinds of different cell-specific tropomyosins around. An interesting little sidelight. Any of you folks have shellfish allergies, like the shrimp or... Okay, the likely candidate, most shellfish allergies are allergies to mollusk and arthropod, like crustacean tropomyosin. They've got tropomyosin as well, but their version of tropomyosin is different enough from the chordate version that it sends some people's immune system wild. So that's actually the major culprit, is the invertebrate version of the very same protein that we have in our bodies. But for some reason, the difference in that protein, for some reason, drives some people's immune systems haywire. They get really nasty allergic reactions, anaphylactic shock in some cases and stuff. And the culprit is, unfortunately, what do you eat when you eat a scallop or a shrimp? What are you eating? You're eating muscle tissue. It's loaded up with tropomyosin. But it's loaded up with the wrong kind of tropomyosin for you folks. Fortunately, I don't have that. We eat like shellfish. The biggest inhibitor of shellfish is the expense. Yeah. Oh, hey, if I get shrimp, if I see shrimp on sale, trust me, I get, I stockpile and they have the 2-for-1 sales, or the really big shrimp for $8 a pound. So, well, I get like a bunch of it, freeze it, and then cook it later. I like shrimp. Okay, anyway, all right. Well, let's take a look at a few things. When you look at myotubes, that's going to be skeletal muscle. And then cells, the fibroblasts, and that's just two specific cells. Fibroblasts are the kinds of cells that produce connective tissue. Recall that myoblasts and fibroblasts are not that different from each other. The precursors are not that different. But one direction, if you have the myo-D proteins expressed, you'll take these generic mesodermal cells and push them down the myoblast to myotube transition. If you don't have that and you have a few other transcription factors, you end up as a fibroblast. And some chemical treatment can actually do things like turning precursor fibroblasts into myoblasts that form perfectly functional myotubes. So, there's a certain decision point where do you go this way or do you go that way? Okay, so they're fairly similar in origin. They came from these precursor mesodermal cells and then diverged later on. And it turns out that all the different versions of tropomyosin are coded for by just one gene. But that gene is alternately spliced. And we have 11 exons. And how they're spliced together. The different cell types have different splicing factors that are either going to inhibit spliceosome formation at some junctions, or they can promote spliceosome formation at intron-exon junctions that ordinarily would not happen. In other words, you have a strong splice site and you block it, or you have a weak splice site and you beef it up. Okay, so we take a look at what goes on here. We have for the myotubes. For the myotubes, we splice everything to, oops, wrong one. Oh, we go up to the pen. We splice all the introns together except we toss out exon 6 and toss out exon 11. We get rid of those two. When we do the fibroblasts, we toss out some different exons. We keep 1 to 6. And we toss out exons 7 and 10 instead. So 2 different splicing patterns caused by 2 different splicing factors that are in these cells, and you get 2 different versions of tropomyosin. One specializes in skeletal muscle, and the other is a more generic thing that deals with the microfilament system in these cells. And there are other cell types that will have somewhat different splicing patterns. With 11 exons, you can make an awful lot of different versions of tropomyosin. But this way we get, out of one gene, multiple cell-specific versions of these proteins. And we do that because somewhere down the line, before we express those genes, we have splicing factors that either promote or inhibit spliceosome formation at certain points, and those splicing factors, which are cell-specific, determine what kind of tropomyosin you're ultimately going to get. So that's alternate splicing in a nutshell. But the coolest example here, which actually requires several different things, where you have alternate splicing of genes that code for splicing factors, and then ultimately ultimate splicing of a gene that leads to 2 different transcription factors that activate 2 different sets of genes. And what this is one of the coolest examples of alternate splicing, multi-step kind of pathway here, is where we look at sex determination in fruit flies. What makes a man if you're a fruit fly? Okay, it's alternate splicing. Okay, the quick and dirty here. You have sort of a hierarchy of genes. The first gene is called sex lethal. I don't know why I got that one, but it's abbreviated SXL. God. Sounds like somebody I knew when I was a student. He had sex with her, you were toast. She dumped me at the most inopportune moment. Okay, all right. Well, okay, well what this thing does is this one codes for a splicing factor. And what it does is a splicing factor for another gene. And then what that other gene is called a transformer gene. Okay, and the transformer gene actually happens to be a splicing factor. Another splicing factor goes for a different gene. Okay, and the transformer gene, the splicing factor from that is going to, yeah.
In your parentheses on the transformer, is that TRA?
TRA, sorry.
Okay.
Uh-huh. Okay. And the transformer splicing factor is going to do alternate splicing of yet a third gene called the double sex gene. Boy, that sounds like fun. Okay. And what the double sex gene codes for is a transcription factor. The end result is we're going to get two different versions of this transcription factor. One activates the male-specific genes, the other activates the female-specific genes. It doesn't touch the male genes. So we have this interesting hierarchy. 3 proteins, 2 of them code for splicing factors that does alternate splicing in the next level down. And then finally you do alternate splicing with transcription factors. Coming in with 2 transcription factors that activate transcription of different genes by binding to a different enhancer. The DNA binding site has been altered by alternate splicing. So that's the quick and dirty. So let's take a little look at a few things and how this thing works. First, let's look at our sex lethal thing. And we're going to look at the case for males and females. Now, the sex lethal codes for a splicing factor. Okay, the gene itself is kind of interesting. It's got 4 exons, but it actually has 2 promoters. In fact, let's do the gene here. Where's that one promoter located? Okay, it's got 4 exons. It has 1 promoter in the typical place of a promoter. And we call this the late promoter because it can be activated with a time delay. Okay, so that's 1 promoter. It's not where you generally find promoter, in front of a gene. But there is a second promoter, and that second promoter is actually located inside one of the introns, and we call this the early promoter. So this guy has 2 promoters. And the question is, what one gets chosen? Okay, well, let's look at the case for the males here. Now, these different, even at the point where we're getting to future males and future females, there are going to be different transcription factors in those organisms. And what happens in the case with males, you have a transcription factor that activates preferentially this late promoter. Okay. So we have, here's our late promoter, and then we have our 4 exons. And here's the early promoter, and we've got. So for the males, the late promoter preferentially gets activated. Now we end up with transcription and splicing, and here's our messenger RNA. We splice all 4 of the exons together. 2, 3, 4. 1, 2, 3, 4. Toss out all the intron sequences, including the one containing the early promoter. So now we get all these things together. But, guess what? There's a little nasty surprise in exon 3. Because partway through exon 3, there is a stop codon. There's another one at the end of exon 4. Normally, stop codons are found at the end of a gene because that tells the ribosome to stop translating, stop hooking amino acids together at this point. This thing has 2 stop codons, one where it belongs and another in the middle of one of the exons. So now when we make the protein, we have parts from exon 1 exon 2, we got exon 1, exon 2, and then we have the part, but not all of exon 3, and none of exon 4 at all. So we're missing part of exon 3, or the sequence from that, part and all of exon 4. End result is this is a shortened protein, and this protein doesn't do jack. So the bottom line with the males, we produce a non-functional we produce a shortened version of a splicing factor that doesn't splice anything because you're missing too much of it. All right, that ain't good, right? So let's take a look at the case for females. Can I erase the generic gene here at this point?
Yes.
Okay, that's just a map of the gene. All right, now let's look at the case in females. Females operate a little bit differently. In females, there's a transcription factor that preferentially allows transcription at the early promoter. And that's what's going to get transcribed first. So, I'll draw the gene here again. LP for late promoter, exon 1, exon 2. Here's our EP, our early promoter. We're going to activate this, exon 3, exon 4. Okay, so that's the gene. We're going to transcribe it first off of the early promoter. So now we get a nuclear RNA consisting of exon 3 and all of exon 4. Okay, we're going to glue these guys together, and we're going to get a small version of the sex-lethal protein. Okay, so we get a shortened version of the sex-lethal protein. Now, what this is going to do, this turns out to be a splicing factor again. Okay. Now, it turns out this small or early sex-lethal protein, that happens to be a splicing factor that splices the RNA precursors of the sex-lethal gene itself. So we're going to have a second step. We got this. Now let's go back to females here. All right. This is still going to be females. This is our next step here. Okay, now we got that small shortened version of the sex-lethal protein. It's a splicing factor for the same RNA. Now what happens? We activate that early promoter first because the females had, or future females had transcription factor that the future males didn't. So we turn on the early promoter first. But now we get the late promoter gets turned on. Okay, but in this case what we're going to have is we're going to have, let's do the nuclear RNA. Actually, it's going to be nuclear RNA. Okay, what we're going to have here is we're going to have our little sex-lethal protein, the early version of it. That's a splicing factor. What it does is it's going to bind to either end of exon 3. Okay? Now, what this is a splicing inhibitor. It blocks spliceosome formation here and here. So instead, in the males, we got spliceosome going from 3 to 4 and from 2 to 3. We don't have that anymore. We cannot form a spliceosome at these two junctions. So instead, we're going to have the spliceosome bridging all the way from 2 to 4. Because here again, strong splice site and the early or little sex-lethal protein blocks that strong splice site, preventing exon 3 from getting spliced in. So now what we end up doing, in this case, is we splice out and discard, exon 3 entirely. So now in our final messenger RNA in females, we have eliminated exon 3 with that nasty little stop codon in it. Yes?
What is the circles on top of exon 3?
Okay, the circles on top of exon 3, SXL, sex-lethal protein. That's the short version that you find generated from the early promoter that you find in females. So the early promoter in females gets activated first, and we splice things so we get a little tiny, we get part of exon 3, exon 4, we miss the stop codon, we get a little tiny version of a protein that is a functioning splicing factor, a different target. It binds here, and it's going to prevent exon 3 from being spliced when we finally get the whole shebang together. So in this case, in the males we have spliced this protein. You look puzzled.
No, I'm just looking at it.
Oh, okay. In the males, the take-home lesson here, the males you splice all 4 exons together, but there's a stop codon in exon 3 that causes premature termination of translation. In other words, we make a short protein that doesn't function. Now the second thing, in females, we get that early protein that actually acts as a specific splicing factor. It causes the elimination of exon 3 entirely. So we splice exon 1, 2, and 4 together, and that makes the functioning sex-lethal protein. So that's the first thing. Bottom line, males do not have a functioning sex-lethal protein. Females do. And these versions of the sex-lethal protein, what they're going to do is act as splicing factors for the transformer gene. And that's our next one. So let's go down to transformer gene here. So we've generated a functioning splicing factor in females, a non-functioning one in males. So our next step here, the transformer gene, this also is a splicing factor, and that's for the double sex gene later on. Okay. We're going to make another film on that. A transformer, yes. It can turn males into females and vice versa, depending on how it's spliced. Hey, that's very much like the film, you know.
Well, you wouldn't have to worry about us turning into anything.
This can
into
turn
male.
all kinds of things, you know. The transformers turn all kinds of things. So in this case, this transformer turns into 2 different splicing factors. And then they turn the whole thing into a male or a female. Okay, well anyway, let's take a look at the transformer gene. Okay, our transformer gene, we happen to have 3 exons. 1, 2. Fortunately, just 1 promoter. So we don't have those complications. Okay, we have 3 exons here. Now, males. Okay, the case for males. The males don't have a functional sex-lethal protein because it gets terminated prematurely thanks to that internal stop codon. So, in other words, nothing's going to get spliced. You splice all you get, or nothing's going to get alternately spliced. So the final messenger RNA here is going to be 1, 2, 3. Exons 1, 2, and 3. And lo and behold, this thing is not functional again. That's interesting. I think about 3/4 of the class would probably agree. It looks like males are full of non-functional things. That's why 3/4 of this class is female. Males are non-functional due to a lack of proper splicing. Okay. All right. Well, we splice everything together here, and spliceosomes form and stuff, and we end up with all three exons spliced together. Unfortunately, it produces a non-functioning protein. Okay, now females are going to be a little bit different because we have the sex-lethal protein, or rather, yeah, the sex-lethal protein is going to block spliceosome formation on one of the junctions on the intron 1, exon 2 boundary. So let's kind of look at this here for the females. Okay. Here's the gene. Well, actually, here's the nuclear RNA. Okay, what we're going to do here is we are going to put, we're going to block this particular boundary right here. Okay? So sex lethal is a splicing inhibitor at this splice site junction. Now what you might expect here is you get a spliceosome formation going all the way from 1 to 3 and bridging going from 1 to 3, and that meant you would eliminate the introns and all of exon 2. But that's not the case, folks. Because here is when a cryptic splice site comes into play. Remember, we say cryptic splice sites are hidden in, usually in exons, occasionally in some introns. They are functioning splice sites, but they're weaker than normal ones. So if you block the normal splice site, then the cryptic site comes into play. If you're a molecular biologist and you deliberately destroy a splice site, that's how they were first discovered, they found that certain RNAs still got spliced, but got spliced differently. And they said, oh, there must be a hidden or cryptic splice site buried somewhere in there. So that's what happens. We have a cryptic splice site here. Remember we mentioned that. That allows you to discard part, but not all of an exon. Or allows you to keep some intron sequences in if you need to do so. So here we have a cryptic splice site. That's another principle we've mentioned here, cryptic splice sites. So now what we're going to get is we're going to get a spliceosome. We're going to get something like this. We're going to get spliceosome forming here and here. Okay, now what we're going to, or rather here and here. So what we end up with in our final messenger RNA is we have exon 1, we have part of exon 2, and all of exon 3. We're missing part of exon 2. The end result is a functioning protein. If you have all of exon 2, that warps the tertiary structure of the protein. I don't know what it does. I don't even know if they have the structural information on that. But it does something to the structure of that protein where it can't bind to splice junctions and things like that. But this version can. So once again, males get the short end of the stick. Let's face it, folks. Let's face you, ladies. We're non-functional. My wife would actually agree with that at times. Okay. All right. God, this guy's really getting the short end of the stick here. Okay, so the bottom line here again. Males produce a non-functioning sex-lethal protein. You don't get splicing of the transformer gene or the transformer RNA. So you get all the exons put together. You get a non-functioning transformer protein. Females, you do get the functional sex-lethal protein. It allows you to discard part of exon 2 and keep the other part because we have a cryptic splice site. And then they get the functional protein. Yes?
Is it supposed to be 3 or exon 4 for the girls?
Oh, 3. We only have 3 exons in this.
Okay, because
Sex lethal is 4.
You skipped 3.
Okay, well we threw out exon 3 in females because that's the one, and that was the sex lethal gene, different gene test. In the sex lethal gene, we threw out all of exon 3, and the reason we did that is that's got a stop codon in it. So we tossed it into the trash. Okay, in this case, the transformer gene, we only have 3 exons. Males splice everything together, but females block the normal splice site and activate a cryptic one. So we keep part of exon 2, discard the other part. Yes?
So cryptic sites are different ones, right?
No, well, in this case, but cryptic
But
sites,
they can be found.
yeah, cryptic splice sites can be found in a large number of genes. It doesn't matter whether you're male or female. This example here uses a cryptic site, but a wide range of genes are going to have cryptic splice sites in them that will be, and it can be one cell type and non-other. It doesn't have anything to do with males or females necessarily. In this example, it actually plays a role. But they can be found in a wide range. Not all genes have cryptic splice sites in them. Most of the time when you block the splice site experimentally, you just didn't get splicing there. But sometimes you did get that splicing by activating the cryptic site, which is how they got discovered. Okay, so it doesn't matter, just the individual genes. Some have cryptic sites, and some exons work, some introns, some don't.
Okay, so it's just kind of random, like, it's not always on the two.
Yeah,
It's
yeah.
like,
A cryptic
it
site could be
could've been on
a
four, it could've been
cryptic
on
site
10.
could be anywhere and in any gene. And the idea behind that's another role of alternate splicing. In this case, rather than throwing a whole exon out, we can keep part of an exon. Or in much rarer cases, we might even deliberately include intron sequence. So generally, we don't want to do that. That often could cause problems. But sometimes some of the stuff, if you read an intron, you may put amino acids into a protein that actually will alter its function, but not screw it up completely. Okay.
I read about the cryptic sites in the like supplement or whatever
Yeah.
you call it. And the thing I didn't understand is if they were like really random or if they're just, if it actually has like a system.
Yeah, I mean, there will be certain, we don't know what the system is,
Oh,
but you could have a gene with, say, 10 introns and 11 exons. You may have no cryptic sites in there at all, but maybe you have a cryptic site in exon 3 and exon 7, but not in any of the others. Or maybe you'll have cryptic site in just one exon, one specific exon. Or maybe you'll have them in 3 or 4 different exons. It just depends. And that allows you to still get functional proteins, but different versions of them. Okay, so it's going to vary from gene to gene and exon to exon. Okay? All right, so we have this. Bottom line, females get stuff that works. Males, forget them. They're non-functional. Some of you ladies probably have had experiences with that, right? Boyfriends, spouses, ex-spouses, things like that. Okay. All right. Okay, so now we've got the transformer RNA. Now let's go to the double sex gene. That sounds like fun. Double sex. Double your pleasure, double your fun. That's a good way of doing it too, folks. Okay, let's see. My wife won't let me do that.
Okay, that is good.
Yeah, yeah, you strict rules. Strict college rules, don't do that kind of stuff. All right. Okay, the double sex gene has 6 exons in it. All right, now, what's going to happen here is we're going to get alternate splicing patterns, and once again, the transformer protein is a splicing factor for the double sex nuclear RNA. Males don't produce a functioning transformer protein. So if we look at the case of males, that's pretty quick and simple here. They simply don't have a functioning splicing factor. So in this case, what's going to happen is we splice. All these splice sites are strong splice sites in this case. Once again, 1, 2, 3, 4, 5, and 6. So all 6 exons are going to get spliced together, and it makes the functioning male version of the double sex protein. And what this one does is bind to enhancers that are located in front of genes that code for male-specific proteins, thereby turning that embryo into male, especially things, major parts of the anatomy. Of course, it's going to be the gonadal development are the major things that we see in the case of males, but a lot of it's related to gonadal development, but also it's going to involve things like the formation of neural pathways. So males, in the case of fruit flies, male fruit flies are going to have certain behavioral patterns where they'll be attracted to female fruit flies and attempt to mate with them. They won't be attracted to male fruit flies. And well, you do sometimes get organisms, and some of the males will, partially to get them out of competition, will act and look like females for the animals, and they will try to mate for them, which of course is unsuccessful. They're losing their sperm, and meanwhile this guy who's been playing a trick on them, okay, he's just gotten rid of the competition effectively because he shot his sperm into something that's biologically non-productive. And then he goes out and he has all the sex he wants, so his gene gets spread. The guy that got deceived, his genes don't get spread. There's lots of interesting sexual tricks in various kinds of organisms. So in this case, we activate the male-specific genes, largely involving gonadal formation, but also involving neuronal wiring and behavioral patterns. Okay, now, in the females, we do have the functioning transformer protein. Okay, so what happens in the case of females? We're going to get a transcription factor again, but what's going to happen here is we are going to oh, sorry, made a little mistake with the male version. Okay, sorry about this. Go back to correction here in the male version. Yeah. Okay. It turns out here, it turns out that the splice junctions at exon 4 are weak. In other words, ordinarily, correct my mistake.
No, I have a question.
Okay.
Now functioning what?
Transformer splicing factor. Okay. That was the previous one. The males did not make the splicing factor, the females did. Okay, now what we got here, this is an example of sorry about my mistake. This is an example of a weak splice site. The splice sites at exon 4 are weak. They will not allow a spliceosome to form there without some assistance. So in the absence of any splicing factor, we're going to get a spliceosome going from 3 to 5. We kick out exon 4. That's what happens in the male. So we splice exons 1, 2, 3, 5, and 6, but not exon 4 in there. Okay, now, in the case with females, what happens is the transformer protein The transformer protein is a splicing activator. So far, we've been seeing splicing inhibitors that block splice sites and prevent spliceosome forming there. In this case, we have a splicing promoter that will allow spliceosomes to form at weak splice sites that otherwise wouldn't happen. So in this case for females, what we're going to do here is we're going to splice all 6 of the exons together. 3, 4, 5, and 6. And we get a transcription factor that binds and activates transcription of female-specific genes. Apparently, exon 4 is going to have part of the DNA binding sequence or another DNA binding sequence. So in other words, these two recognize different enhancers. One in front of the male genes, another in front of the female genes. And by tossing out exon 4, that determines what enhancers these things will bind to. Okay, so here we have an example. In this case, splicing, alternate splicing, leading to 2 different transcription factors that recognize different enhancers. That's a cool thing about that. Now, the other thing is this is another good example, in this case, of a splicing promoter, promoting spliceosome formation at a weak splice site. All the other things we've seen in this whole chain here were splicing inhibitors that would block spliceosome formation at strong splice sites. So this shows it here. This shows three principles. This whole example here shows three big principles. Principle number one, splicing factors can promote or inhibit spliceosome formation. You block spliceosome formation at strong splice sites, those are splicing inhibitors. But you have splicing promoters that will allow spliceosome formation at weak splice sites. So the default in this case is spliceosomes don't form there until you have that splicing factor. So it's kind of like, in effect, an analogy with transcription. We have transcription factors that promote the basal complex or stabilize it, and you have transcription factors that block or disrupt the basal complex. So that's one point. Second point, we saw an example of a cryptic splice site getting activated when you block the normal stronger one. Cryptic splice sites, in order to work, have to be weaker than the normal splice site. Otherwise, you're going to get preferentially used. So you have to have a strong splice site and a weaker one, which is the cryptic splice site. If the strong one don't work, then the cryptic site comes in play. If the strong one does work, the cryptic site won't get used. So those are two principles that we saw in this kind of example. An interesting way of determining what sex a fruit fly embryo is going to become. Well, that's an example of alternate splicing in making the choice between males and females. Okay, so that was just a couple examples of alternate splicing, and now we're going to go on to something complete. Oh boy.